like to echo the comments of the other speakers and thank the organizers for a fantastic meeting. I also would like to welcome you to the Max Planck family, and that welcome comes from, believe it or not, your nearest neighbor institute, the Max Planck Florida Institute. And I know, in having talked to those of you who come from Germany, it is indeed nearer. The distance to Germany in miles here, 7,000. Distance to Florida, considerably less. And I might also mention that there are some other differences between Germany and the Institute in Florida, certain amenities that you won't find associated with the Max Planck Institutes in Germany, and we look forward to building bridges with the Institute here in Argentina. Now, I'm going to focus today on work that we've been doing for a number of years that looks at the structure, function, and development of circuits in cerebral cortex, and in particular in the visual cortex. And given time limitations, today we'll focus just on the work that has explored the role of experience in the maturation of circuits in visual cortex. And it's worth backing up and just making sure that everyone is on board with the idea that there are a number of mechanisms that contribute to the development of cortical circuits, activity-independent mechanisms, mechanisms that are involve the execution of genetic programs that lead to differentiation, migration, and axon guidance. And then activity-dependent mechanisms. You've already been hearing about these from Tobias Bonhoeffer. Mechanisms that are involved in the formation and selection of appropriate synaptic connections. Now, these activity-dependent mechanisms can be driven by endogenous patterns of activity, or experience-driven activity, and it's the experience-driven activity mechanisms that we want to focus on today. And it's worth emphasizing the importance of experience in the maturation, the early development of cortical circuits. And I think the title of this slide says it best. Most cortical synapses form at a time when experience can influence neural activity. Now, from a structural perspective, what you see here is just the tremendous change in the dendritic morphology of neurons during the early years of life in the human infant, from the rather sparse dendrites found at birth to this elaborate, detailed dendritic arborization that you see 2 years of age. And that's accompanied, of course, by a tremendous increase in the density of synapses in the cortex. And these synapses are derived both from excitatory neurons and from inhibitory neurons. So the bottom line is, at birth, cortex is tremendously immature, and there's tremendous structural rearrangements that occur early in development that are important for cortical function. Now, these changes in structural features, of course, must be associated with changes in the response properties of cortical neurons. And it's worth asking, what are the properties that distinguish neurons in the cortex from antecedent levels in the visual pathway? And this is work that was done a number of years ago by Hubel and Wiesel. And one of the properties that they identified was selectivity for orientation of edges. So if you record from neurons in the visual cortex of animals and present edges at different orientations, you find that there is a preferred edge orientation that's most effective in driving the responses of cortical neurons. Likewise, Hubel and Wiesel demonstrated another property that distinguished neurons in the cortex from earlier levels, and that is selectivity for the direction of stimulus motion. That is to say, these neurons are sensitive to the sequence of motion of visual stimuli, preferring one direction over the opposite direction. And the third property that emerges at the level of cortex is one that Tobias Bonhoeffer has emphasized and has actually been the aspect of cortical development that's been studied most, and that's this issue of binocularity, the fact that it's the first point along the visual pathway where the inputs from the two eyes are brought together, and Tobias has discussed a lot of the work that's been done trying to understand the plasticity that's associated with that process. Now, in many species, and this is also work that was done initially by Hubert Wiesel. Must be a fan of Hubert Wiesel, clearly. Demonstrated that there was a columnar architecture to cortical response properties. So if you stick an electrode vertically into the cortex, almost all the cells you encounter have roughly the same orientation preference. If you run that electrode tangentially, you find, at least in some cases, an orderly progression of orientation preference. And the availability of intrinsic signal optical imaging techniques made it possible for the first time for us to visualize these representations. Here, it shows you the pattern of activity that it's visualized in the cortex of a ferret. Dark regions represent regions that are activated by the visual stimulus. Light regions are not activated. And the beauty of this technique is that you can present a broad range of stimuli, here different orientations, see how the cortex is activated, and literally reconstruct the map of this particular stimulus property. Now, orientation is not the only feature that's mapped in a columnar way. It turns out that selectivity for direction of motion is also mapped in a columnar fashion. And indeed, if you look at a piece of cortex, and just to give you an idea of the scale here, the distance between these domains is about 1 millimeter. Here's a region of cortex. Again, this is a bird's eye view. You're looking down on the surface. This is the map of orientation. Within that map is a nested map of direction. So for example, this region prefers horizontal and has separate regions that prefer opposite directions of motion. Superimposed on that is a mapping of ocular dominance. These are the ocular dominance columns that would actually be tessellating this whole region. And finally, I haven't mentioned it, but I'm sure most all of you are aware, in addition to those maps, there's an orderly mapping of visual space. And so for us, it's been very interesting not only to understand what these maps are and what they do, but what the mechanisms are that are responsible for their development. And it's worth just taking one moment to make the point that the maps that I'm showing you and the patterns that I'm showing you will be found in carnivores and primates, but they would not be found in this critter, the mouse. And I have to say, much like the Tom and Jerry cartoon, many of us are tremendously envious of the mouse toolbox, but we think it's worth asking whether the rules that have been discovered in the mouse apply or generalize to cortices that have this more intricate organizational structure. So this shows you the timeline of the development of maps in the ferret. Ferret is born in a relatively immature state. Its eyes actually open at P30. Here you can see some of the significant points in the development of visual responses. And what you see here is the fact that ocular dominance columns and orientation columns arise prior to eye opening. So very clearly, those properties emerge in the absence of visual experience. But what we found was that direction columns are actually unique in this regard. They arise after the onset of visual experience. If animals are dark-reared, during the period of time when direction columns normally emerge, they fail to emerge, and activity given later on in life, when the animal has been deprived during this critical period for direction, these animals do not develop direction selective responses. It's worth just pointing out for a second that this is a very early in the experience of the animal. much earlier than the critical period for ocular dominance plasticity that Tobias Bonhoeffer has been mentioning. So while there's been tremendous focus on ocular dominance columns and the plasticity associated with that, there's also a lot going on in this so-called pre-critical period that's important for the maturation of other response properties. Now, just showing that these response properties do not emerge in the dark doesn't really say much about whether visual experience or how visual experience plays a role in their emergence. And so what you'd like to know is, does experience with visual motion drive the emergence of direction-selective responses? And we discovered that we could actually visualize the emergence of direction-selective responses in visually naive animals by presenting them with moving visual stimuli. So here's an animal that's been set up, anesthetized, paralyzed, looking at this video monitor. A stimulus is moving back and forth on the screen. When we start the experiment, there's a map of orientation, but there's no sign of direction signal. And then we begin the process of training. So half-hour sessions where the animal experiences a moving stimulus, half-hour no moving stimulus. And this is what we found. So initially, not much happening, but then out to about 8 to 10 hours, we begin to see something that looks quite like a direction signal. Now, it doesn't look as nice as what you see in a mature map, but this is also relatively short duration of experience. What we wanted to do was to see if we could connect what we saw in the cortex with the stimulus that the animal had been visualizing. And we did this at the end of the experiment. What we could do is to present all directions of motion to see whether we saw the emergence of direction-selective responses. And as it turns out, we only saw the emergence of direction-selective responses for the directions in the training stimulus. This animal was trained with a stimulus moving up and down. This one was trained with a stimulus moving left and right. So very clearly, there's a specific training effect here, neurons becoming selective for the direction of stimulus motion. I might also say that we were very happy to see this because this also says that these effects can't be explained by something that's happening lower in the visual pathway. This says that it must be some change that's occurring at the level of cortical circuits. Now, the intrinsic signal imaging is nice. It tells us that columns are emerging, but what you would like to know is what's happening at the level of single neurons. And for this, we turn to 2-photon in vivo imaging of calcium signals with Oregon Green Bapta, and this has also been introduced by Tobias Bonhoeffer. And it allows us to literally go in and look at a cellular level at the factors that are responsible for the emergence of these columns. And so what we can do on a cell-by-cell basis is go in and measure the response to different visual stimuli. And what we find is some cells, and what's on this axis here, I'm sure you can't see it, is 360 degrees of visual space. Some cells respond to only one direction of motion, and some cells respond equally well to both directions of motion. And we developed an index for this. And what I've done here is color code things. So cells that have an index value greater than 0.5 were called direction selective, and this is commonly used in the literature. Those that have less than a 0.5 DSI are orientation selective, but not direction selective. And so the first question we asked is, do we see differences in the direction selectivity of individual neurons consistent with what we saw with intrinsic imaging? And this just shows two different animals, an animal that had 1 day of experience, an animal that had 7 days of experience. Quite striking change in the selectivity for direction of motion, and this is the quantification for a number of animals. Now, what one would like to do is to be able to track individual neurons over time to see whether and how they acquire direction selectivity. And these experiments, for those of you who haven't used OGB, Oregon Green BAP to do this, these were non-trivial experiments, and the graduate student and postdoc involved in this work deserve an enormous amount of credit for getting it to go. What they were able to do is define cells, identical cells before and after training, and see in these neurons a striking change in their response, going from cells that were equally responsive to two opposite directions of motion to being selective for one of those directions of motion. And this just illustrates one field of view before and after training, showing the tremendous increase in direction selectivity. Now, to sum up a lot of work that went into this, this shows the direction selective indices for cells. This is a cumulative percentage gram. And this is looking at experienced animals. And you can see you have a number of cells that are beyond the 0.5 direction selective index. These are the cells from visually naive animals. And this shows that we have a significant effect of our training, even though our training is for a relatively short period of time. And finally, you might ask, is it really motion, or is it just visual stimulation? So to test this, we took the same stimuli, but instead of drifting them, we flashed the stimuli. And when you flash the stimulus, instead of drifting it, so there's no motion information, you don't see the change. Now, some of you may be saying to yourself, wait a minute, there's a problem here. The stimulus that's being presented is moving in two directions. And yet these cells are choosing one direction over another. How is this coming about? Well, we reasoned that perhaps there is a weak initial bias that experience builds upon. And so we went in to see if we could pull this out. We changed how we were quantifying things, and we're displaying now the response of these neurons. So the arrow indicates the preferred direction. The size of the arrow represents the magnitude of that preference, how different the response is to the two directions of motion. This shows you what the pattern that you see in an experienced animal. Note big arrows. Arrows for neurons adjacent to each other are all pointing in the same direction, exactly what you expect for a columnar representation. This is what you see in a visually naive animal. You find short arrows, relatively small difference between the two directions of motion, and you see cells sitting next to each other that have very different direction preference. Here we've coded it blue and yellow to make it easier to see. So what we wanted to know is whether there was some information in visually naive animals upon which experience was building. And so to evaluate this, we used what we called a local coherence index, which just looks at the preference of neurons around a given cell. If they're all pointing in the same direction, that would be plus 100 coherence. If the cell is singing its own tune, very different from its neighbors, then that would be negative 100. And if they're about equally balanced, it would be a local coherence index of 0. And what we found when we looked at local coherence versus distance, for mature animals, we saw a drop in local coherence with distance, exactly what you would expect for a map. But we also found